Hello, welcome students. We are studying the DNA. Although we have studied so many points on this DNA, so today I think we shall see the salient features of DNA. Salient features of DNA. What are the? You know that DNA is a polymer of nucleotides. Means it contains so many molecules of the nucleotides. Understands? And the second, it is a double helical structure. Double helical structure, and you already know that nucleic acid was first discovered by Frederick Michel from the pus cell, and he called it nuclein, and the term DNA was first used by Jacques. He first used the terms DNA, and the model, model was given by Watson and Crick. This is the first. It was nucleic acid discovered by Frederick Michel. The term DNA was used by Jacques, and the model was given by Watson and Crick. So polymer is a polymer of nucleotides. Poly means many nucleotides molecules are combined. We have already discussed. It contains the sugar, nitrogenous base, and phosphoric acid. These are the three main constituents of the DNA. In the sugars, that will be deoxyribose, a 5C compound, deoxyribose. Then here is, we have already studied that the adenine, thymine, guanine, cytosine, these are the bases which are nitrogenous bases. They are attached with each other by hydrogen bond, and these nitrogenous bases attached with deoxyribose, okay, and these are attached by phosphoric acid or phosphate. So these two means sugar and phosphates are the backbone of DNA. These two are the backbone of the DNA. Nitrogenous bases are attached with each other by hydrogen bond. They are the hydrogen bond. We see that 2 hydrogen bond between A and T and 3 hydrogen bonds between G and C. Understand? The length of each turn is 34 angstrom. This from this is a complete turn from this point to this point. This is its length is 34 angstrom. Understands? Or you may also tell that 3.4 nanometer. That is 3.4 nanometer. In each turn, 10 base pairs are found. So, from this to this, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 base pairs are found. So, the distance between two nucleotides is 3.4 angstrom or 0.34 nanometer. So that is 34 by 10 or 3.4 by 10. That is, each has a distance of from this to this, each a distance of 3.4 angstrom. Understand? Now these are the salient features of that. One thing, unite there. A, T, G, C, there is a basis. I have already that there's a valence and there's a pyrimidine. Now the DNA has bases A, T, G, C, that is adenine, thymine, guanine, cytosine. These two are called purines and these are pyrimidines, as I have already told you, and they make their base pairs by means of hydrogen bond that we have already discussed. So we can say that nucleotide is composed of nucleoside plus phosphoric acid, and this nucleoside is composed of sugar plus nitrogenous base. So the DNA contains nucleotide, that is nucleoside plus phosphate, and this nucleoside, this one. So and so many nucleotides combines to forms DNA, and it is a double helical structure, and each turn contain, and you already know that the diameter is 20 angstrom, means that is this to this one, this differences is 20 angstrom. Now these are the some, and you know that DNA is found in the nucleus on the chromosomes. What means found in nucleus on chromosomes, and it is acidic in nature. It is acidic in nature, and because it is found in nucleus and acidic in nature, it is called न्यूक्लिक एसिड. चूंकि ये न्यूक्लियस में पाया जाता है और इसकी स्वभाव एसिडिक होती है, इसलिए इसको हम न्यूक्लिक एसिड के नाम से जानते हैं. या फ्रेडरिक मेशर ने इसी कॉज पे, यानी बिकॉज इट इज फाउंड इन न्यूक्लियस एंड एसिडिक इन नेचर, ही कॉल्ड इट एज न्यूक्लिक एसिड. नाउ द model was given by Watson and Crick, and the Crick also states a central dogma. A central dogma. We know that it is a nucleus, it is the chromosomes, a DNA is found, and we also know the importance of protein in the body is must, and that protein decided the design of the body and all the other activities, and that protein is synthesized on the ribosome. Protein is synthesized on ribosome, and what protein will be formed, that information is given by DNA. That information is given by DNA. यानी DNA information देता है, समझे, कि किस टाइप के प्रोटीन्स बनना है, वो प्रोटीन वहां बनता है. And that is so how it is found in nucleus, it is found in cytoplasm. How this message goes to the ribosome, that is by means of ribonucleic acid. That is by means of ribonucleic, means DNA जो भी मैसेज देता है by means of messenger RNA, उसको हम बाद में चलेंगे. RNA के टाइप्स होते हैं, समझे, वो ये एक टाइप्स है, समझे. तो ये टाइप्स, ये मैसेज को यहाँ से राइबोसोम तक ले जाता है, समझे. तो the DNA, RNA, this is protein. This process by which the DNA changes into RNA, that is known as transcription, and ribonucleic acid to protein is called translation. We will discuss in the next video about that. And the DNA itself replicate itself, that is by means of replication, it by means of it replication, it forms another boundaries, another helix of DNA. So this process, this idea or this structure is known as central dogma. It is given by Frederick Crick. It is given by Crick. So this is about something salient features of the DNA. Now the next, that is packaging of DNA. How the DNA packaged, how the DNA made the helix coiling structures, how they are attached. So actually we know that it is made up of double helix, and it is called, the distance is 3.4 nanometer, that is 35 angstrom. In this there are 10 nucleotides. It contains 10 base pairs. So the distance between two base pair is 0.34 nanometer on this. And how they arranged. And second, in a general mammalian, about 6.6 into 10 to the power 9 base pairs are found. About the 6.69 into 6.6 into 10 to the power 9 base pairs are found, and the difference between two base pair is 0.34. So we can say the length of DNA is 0.34 into 10 to the power minus 9 into 6.6 into 10 to the power 9. Understand? This will be cancelled. So about this means 2.2 meter. So the length of the DNA is 2.2 meter. And like that, if the question is asked, if someone has 5.5 into 10 to the power 6 or 9 whatever, the nucleotides base pair, what will be the length of that? So multiply this with this number. अगर पूछा जाए कि किसी में बेस पेयर की संख्या इतनी है तो उसकी डीएनए की लेंथ क्या होगी, तो उसको हम लोग दो के बीच का जो डिस्टेंस होता है 0.34 एंगस्ट्रॉम, उसको हम उससे मल्टीप्लाई कर देते हैं, समझा? तो दिस इज द लेंथ ऑफ डीएनए. नाउ तो अच्छा, नाउ वी से हाउ द डीएनए स्टैंड्स इज a double helical structures. Double helical structures and is negative charged, and it is, and DNA is negative charge, so it will binds, binds those proteins which is positive charged. तो ये ऐसे प्रोटीन के चारों तरफ बनेगा, वहां एक्यूमुलेट होगा, हम जो नेगेटिव पॉजिटिव चार्ज हो. सपोज इट इज द प्रोटीन हैविंग पॉजिटिव चार्ज, सो एंड डीएनए is negative charge, so it will attach towards itself, it is packaged toward that, it will bind towards that. So and that packaging takes place in both type of nucleotides, that is in prokaryotes, in the eukaryotes. In the prokaryotes, nucleus ill-developed, means nuclear membrane is not found, but DNA is not in scattered forms. DNA is not found in scattered form. अंडरस्टैंड, लेकिन डीएनए स्कैटर्ड फॉर्म्स में बाकी सब तो स्कैटर्ड में पाए जाते हैं, लेकिन डीएनए नहीं पाता। इट बाइंड्स एनी पॉजिटिव चार्ज्ड प्रोटीन। It was because nucleic DNA is negative charge, so it binds around the positive charge protein, and that's why it is not found in scattered form, and this is called nucleoids, means plus a positive charge protein, and this is the DNA negative charged. Understand? So it is known as nucleoids. But in the eukaryotes, the situation is somewhat different. That DNA is packaged in found around the histones. Means protein histones are the responsible, and this protein histone is a positive charged. Protein histone is a positive charge, and the charge of any protein is depend upon the amino acids, and because its positive charge is due to lysine plus arginine. Due to this, its positive charge are maintained, and if it presents in octet form, यानी 8 हिस्टोन्स एक साथ मिले होते हैं, लाइक दैट। These are the histones. So because they have been, so this is called histones octamer, means 8 histones in a bundle, and the amino acids and the DNA is attached to that double structures. DNA is a negative charge, it is positive charge, and it is negative, so it attaches in that, and this is known as nucleosome, and depending on the binding, it forms two types of, this is a chromatin molecules, two types of chromatin, euchromatin and heterochromatin. They are loosely arranged. They are densely arranged. This is active and this is inactive. Loosely arranged, so they take light stain and they take dark deep stains. So the two types of chromatins on that base. One is loosely arranged, so it is called euchromatin. One is densely arranged, known as heterochromatin. It takes light color, it takes dark color, it is active in nature, it is low active or inactive in nature. So that is the salient features and the packaging of DNA.